Here we present a protocol used to examine the effects on levels of biophysical and psychological stress, following visitation to 3 locations with different levels of nature. We're interested to learn whether a site with higher levels of naturalness would be more effective in helping participants to reduce their stress levels. In addition to using self-report questionnaire, the main advantage of using this technique is that analyzing levels of cortisol and amylase collected from participants' saliva samples can give us a more accurate read and better understanding of how our body actually responds after visitation to recreation sites. Demonstrating the procedure will be Dr. Hocevar, a professor from Indiana University.
Begin by introducing yourself to the subject. Provide them with a consent form to sign, explaining the voluntary nature, purpose, and procedures of the study. Give the subject a red armband for future identification. Obtain physiological and psychological measures of stress levels via saliva samples just prior to the recreational experience and immediately following conclusion of the experience. Collect a 1 to 2 milliliter saliva sample using a passive drool method. Instruct subjects to allow saliva to pool in their mouths, then drool down the 2-inch straw and into the cryovial until 1 milliliter has been collected. Label samples with an assigned 3-digit ID number and letter to indicate the timing of data collection. Then, temporarily freeze the sample in a steroid foam box full of dry ice for no more than 2 hours. Transport the marked samples to a laboratory.